Good morning, Allison. How are you doing today?
I'm doing great. How are you?
I'm doing fantastic, thanks. Hey, I was wondering, do you think you could share with us the PhD thesis that you brought with you this year?
Yes, I would love to. So my research is actually over the human skin microbiome as an alternative source of DNA to identify evidence from crime scenes. So obviously we're not quite to the collecting evidence from crime scenes part of it, but we really hope that this will be a good new marker system for human identification. So my project is focusing on samples or skin samples that were taken from individuals and looking at whether we can accurately predict which individual those skin samples came from.
Wow.
So we took 3 replicate swabs on either the hand, the foot, or the manubrium, which is up here on your collarbone. And then previous work had shown our classification accuracies for those samples using particular algorithms was about 70 to 80%. Hopefully, I'm going to get that a lot closer to 100%. So initially, I was looking at samples from the same individuals. So we consider their microbiome on them as a population. So think about it as the populations we use in forensics. We're talking about the same sort of thing, but now we're talking about microbes and on a single person. We're looking at replicate 1 versus replicate 2 or some other replicate on the same individual and seeing that those two are consistent. Then I went on to compare what we were calling incorrect or not associated individuals.
Okay.
So these are 2 different individuals that are being compared against each other. So if it's incorrect, that means in a previous study we showed that maybe individual 1 classified as individual 5. Well, obviously that's not true. The sample individual 1 because we know. So I'm trying to look to see if my method better identifies individual 1 as individual 1 instead of whoever they were misclassified as.
Okay.
The not associated is an individual that was correctly identified in all previous algorithms that have been tried with these samples. And so far, looking at Wright's Fixation Index, or FST, which most of our population geneticists here are pretty familiar with, we're starting to see pretty good trends for FST values. So our red lines here are samples from the same individuals. So we have low FST, which means their populations look very similar. And then in our not associated or incorrect individuals, we start seeing higher FST values going out here to the right, which means that their populations do not look similar to each other, which is exactly what we want to see. So far, all good. It looks like my PhD thesis is working, so that's always very exciting to not have to go back to the drawing board. And then hopefully next year I'll be back with a lot more information for our poster and maybe a better marker system too.
That's fantastic. So what you're doing here, is this all a computer model? And where did you get your information from that you used for this?
So the samples are real samples. They were collected by another PhD student that worked on this project before me, and she the marker design for our MPS marker system. We call it the HID Skinplex.
Okay.
But now for me, it is all just data analysis. So I luckily had other students that had to do all the bench work side of this and now just have piles and piles and piles of files to work with. So it's been a great experience.
Well, that's fantastic. Thank you so much for taking time to present for us.
Yeah, terrific.
We'll see you later.
Talk to you again.
Thanks.